Okay, guys, this is lecture 4.7. This is all about regulating the cell cycle. And my encouragement to you is actually to do the POGL first and then come back and take these notes. The POGL, remember, is meant to be a little more exploratory. So done when you don't have actual content knowledge necessarily. That helps us reinforce the skills of analyzing models that we've maybe never seen before. So I would say do the POGL first and then come back and take these notes. And then if there was anything on the POGL that didn't make sense, the lecture might reinforce that. And you could go back and, like, if you had to skip a question, you could go back and answer that. So Section 4 in our notes, regulating the cell cycle. So we were introduced to this as the cell cycle, right? We've all had a little bit of a lecture. Some of us have had some class discussion on it this week. And so you have, when it comes to the cell cycle, you have interphase, which is made up of G1, S, and G2. And then you have the M phase, which is the division component, right? Nuclear division and cytoplasmic division. So in checkpoints, we have these moments that regulate the cell cycle, and they are called checkpoints. And so you were introduced to 3 checkpoints in your POGL. The first checkpoint that a cell would encounter is in G1. So the very beginning of interphase, it is growing and doubling in size. It's increasing its organelle count, and it hits the end of G1, and it's almost like a stop sign. So the cell is paused for a moment at the end of G1, and this is also called the restriction checkpoint. It's the only one that has a specialized name because of what happens here. So as a cell cruises towards the end of G1, there are a couple of things that it really should have done. And if it hasn't done those things, there are a couple of options. So it approaches the checkpoint, the cell approaches the checkpoint, and if it hasn't met the criteria, it can actually stay in G1 until it does. This is the only checkpoint where staying put and continuing to develop whatever is necessary is available. All of the other checkpoints, if you reach them, and you do not meet the criteria, the cell is triggered into apoptosis, which is cell death. So basically, this is the only checkpoint where you get a chance to kind of do over, right? So you could approach the G1 checkpoint and you don't meet criteria, and so you can stay put in G1 and continue to grow. You could also approach the G1 checkpoint and be ejected into G0, which, remember, is that sort of quiet resting state where the cell is still alive and metabolically active, but it is not actively attempting to divide. So this is the checkpoint at which a cell could move into and out of G0. Or it could move forward because all of its criteria have been met. So basically, when you hit the G1 checkpoint, we're looking to see, did we grow as a cell? Did we accomplish the tasks involved in G1? And let's take a peek at our DNA and make sure that we don't see any mutations, anything that would prevent the cell from being copied, or making sure that the cell is healthy enough to be copied. If the answer is yes, once we hit that checkpoint, yes, we've done everything we're supposed to, and we are the kind of cell that should move forward, move on into S. If we are not, we can stay there and kind of try again. And again, this is also the point at which you can move into G0 if you are a cell that moves into G0. The next checkpoint is all the way at the end of interphase, at the end of G2. And so here we are looking at the most significant thing that has occurred. We are looking to see if our DNA really did duplicate correctly in the S phase. So at the end of G2, we're checking our DNA. If your DNA duplicated, you move forward. If your DNA did not duplicate correctly, the cell triggers its own death. There isn't really a second chance here. Apoptosis will be triggered. So let's pretend the cell is fine and it has duplicated its DNA correctly with minimal errors, and it is allowed to move forward. The next and last checkpoint that it will hit is in the M phase. Specifically, this is in metaphase of either mitosis or meiosis, the nuclear division. And what we're checking here is the spindle formation and attachment. So we want to make sure that when mitosis really is fully complete, that the chromosomes have been pulled apart equally. And so this checkpoint is checking the mechanism that will pull them apart, that thing called the spindle that you have seen in a previous lecture, or if you were in class with me this past week, we even talked about it. So check spindle formation. If everything is good, move forward through this last checkpoint and finish out division. And so the idea here is we don't just replicate cells without having a little bit of checks and balance, a system of checks and balances. There are these checkpoints that exist within certain pockets of the cell cycle. And as a cell approaches that checkpoint, there's something that's being evaluated. If the cell meets the criteria, it moves forward. If it doesn't meet the criteria, in most of these, it's going to trigger apoptosis, except for that G1 checkpoint, which is kind of given a second chance. Now, your brain might be thinking, well, Miss Mack, what are you talking about? They approach a checkpoint. Like, these are not, cells are not driving cars. We're not approaching a stoplight or a stop sign. How would they even approach a checkpoint? That comes from Model 2 in your POGIL. So everything you've just seen on this screen was addressed in Model 1. Model 2 talks about something called cyclins and CDK molecules. So the creation of the formation of cyclins binding to their CDKs, rising and declining levels of these proteins are what trigger the cell to kind of pause in development and allow for the assessment at these checkpoints. So cyclins and CDKs were addressed in model number 2. So here's what we need to know about cyclins and CDKs. Both are proteins that control the checkpoints, okay? Cyclins, and this is this section of the chart right here. Cyclins are proteins that the levels increase and decrease as the cell moves through the cell cycle. So their numbers fluctuate. We build them up and then we break them down, okay? They bind to specific cyclin-dependent kinase molecules. Now, we've seen kinases before. Remember, kinases are enzymes that will phosphorylate, add phosphate groups to other proteins in the cell communication pathway to activate them. And so the cyclins essentially are activating the proteins that will go about activating other proteins. So cyclin levels, there are lots of different kinds of cyclins. Usually they're given letters, cyclin A, B, C, D, and those levels rise and fall within the overall cell cycle. And so as they rise, let's say, for example, cyclin D levels increase, they will find and bind to their specific CDK molecules. And once they are bound together, they're considered to be an active complex that will trigger some of the actions occurring at the checkpoints. So we'll see levels rise as we approach a checkpoint of certain cyclins, and then we'll see levels fall as the cell progresses through those checkpoints. The connection that we'll make back to even cancer and how cancerous cells don't really follow these cell cycle regulation checkpoints is this last bullet right here on cyclin. So I'm going to come back to oncogenes and proto-oncogenes. But what you should know is that most cyclins are produced based on the genetic coding in these proto-oncogenes. Okay, it's the name of a gene, and we'll see what happens to that gene that connects it to cancer, which ultimately connects it back to the production of cyclins and how that disrupts regulation of the cell cycle. Let's talk for just, this is a picture of a little cyclin right here. Here is that cyclin's kinase. So remember, kinase is an enzyme. ASE tells us, the end of the word ASE tells us that it's an enzyme. So here is a kinase enzyme that needs to kind of become active before it can go about doing its job. And so that will become active when its specific cyclin binds to it. Once it is activated by its cyclin, we would refer to this entire thing as a complex. And so complexes are really regulating the cell's progression towards checkpoints, the cell's pause at a checkpoint, and even what might happen if the cell does or doesn't meet the criteria of the checkpoint, the actual work that is needed to, for example, trigger apoptosis, or the work that is needed to trigger moving from metaphase into anaphase is going to be done by these complexes, the cyclins attached to their CDKs. So these are little proteins in the cell that will regulate those 3 checkpoints that we talked about. Now, these are really complicated diagrams, but let's talk about other things that might control whether or not a cell is going to divide. So these are non-cyclin controls. All right, so these are usually, these deal with typically things in the external environment of the cell, whereas the cyclins that we saw just a second ago, those are proteins that are created inside of a cell that regulate the cell's cycle. These are things that are impacting the cell cycle from the outside of the cell. So the first non-cyclin control is the use of growth factors. And that's where, I mean, you can see in the word, a growth factor is typically a hormone, but as its name indicates, it's going to induce cell growth and division. And so here are 2 really complex diagrams, but I want you to find the ligand in each of them. Okay, so finding the ligand means finding the receptor that the ligand might bind to. So in this diagram, here's our membrane. Here's a couple of receptors. And this little molecule right here, PDGF, or PD on one, GF on the other, collectively PDGF, there is one ligand that is a growth factor. Here is another cell, membrane around, receptor. Here's the other ligand, VEGF. What do PDGF and VEGF have in common? The letters G and F. So G, F, these are both growth factors. So these are typically proteins or hormones that are released in the extracellular environment that will bind to, that shouldn't say bond, it should say bind to cells at their receptor, right? So these are ligands that bind to a receptor, and that binding triggers a cell communication process that is complicated, but ultimately leads to mitosis. So if we follow this first diagram up here, PDGF binds to its receptor, and it triggers a whole bunch of these internal proteins. And we're not going to go through exactly the pathway, but let's follow this arrow over here. So PDGF binds to its R receptor. It triggers this red protein that leads to something happening over here that leads to, you guys hear my puppy, that leads to this pathway all the way down to this word right here, cell proliferation. That's a big fancy word for the cell divides. To proliferate means you make more. of yourself. Okay, so PDGF binding causes this entire cell communication pathway leading to the response of dividing, the cell dividing. Down here, if we look at VEGF binding to the receptor, cell communication pathway enters the nucleus, some stuff happens, and what do we get? Something called angiogenesis. And there's our word again, proliferation. So cells are dividing. So great examples of these growth factors are the two that I've just showed you. PDGF stands for platelet-derived growth factor. When we were talking about in class, when you get injured and the clotting response kicks into gear, you were introduced to platelets. Platelets help to form a clot. PDGF is a growth factor associated with platelet cells. And platelet cells they need to be increased. Their count needs to be increased if we are repairing something, if we have injury, damaged tissue, we need to build a clot. That's all going to be connected back to needing to make more platelets, which is triggered in this pathway right here via platelet-derived growth factor, an external ligand that causes an internal response that triggers mitosis. This one, VEGF, this is more connected to the growth of vessels, blood vessels, which will supply tissue with blood. And so I believe it's vascular endothelial growth factor is the name of it, is the VEGF. And that causes the cells in your vessels to go through mitosis to build more vessels so that you can stretch blood supply into tissues that might need it. So growth factors are an example of non-cyclin controls, external control that when released causes ultimately mitosis, the cell cycle to kick into gear. Another non-cycling control is called density-dependent inhibition. And this comes from the idea that if cells grow, so let's look at this diagram. You have a few cells. Those cells are going to trigger mitosis until they have grown into a sheet that fully covers this petri dish. Now they're very densely packed here, and so these cells would stop their cell division process because they are densely packed into a space. And part of this is about resource availability. So if there's limited space for cells to grow, if they're already densely packed into an area, they don't want to create more of themselves to eat up some of the limited resources, right? So basically, if we're already crowded in an area and consuming the resources, why add more mouths to the area that will then have to be fed? So if we follow this diagram down, they've done experiment studies that in a situation like this, all of these cells have stopped their cell cycle. But if we come in and dig out a little hole and free up some space, guess what? The cells right next to this space will trigger mitosis to kind of fill in the gap. And so these are this process of creating new cells by way of mitosis that is triggered by proteins that are created based off of tumor suppressor genes. So bank that in the back of your mind because I'm going to come back and connect that to cancer. Okay, so basically, if the cells have crowded an area, they're densely crowded within an area, those cells will turn off their cell cycle and they will not go through mitosis and add more cells to the space. However, if something occurs that frees up some space, they will trigger mitosis in the surrounding cells to fill in that space. So density-dependent inhibition, it's kind of a touch factor, right? So if the cells are touching each other, they stop growing. And if they are not touching each other, they know that there is space to fill in. And the last is very similar to density dependent. This is called anchorage dependent inhibition. This is the idea that cells have to be anchored to a matrix. They have to have access to resources and nutrients for them to divide. And so if they ever start to kind of stack up and they cannot access their matrix, they will turn off their cell cycle. So if they cannot anchor to a nutrient source, then they stop dividing. This is something that cancer cells, tumor cells specifically, violate because they can both increase in their area. So they sort of ignore the density dependent stuff that we talked about a second ago, and they accumulate one on top of the other. So they ignore this anchorage dependent inhibition as well. And so again, we'll try to connect those back to cancer. But those, really, those three things are ways that the cell cycle is regulated that doesn't involve cyclins. So we have to learn about cyclins. Those are internal control mechanisms. But then we also need to know about these external control mechanisms for regulating the cell cycle as well. All right, you are done with this section of notes.